On behalf of our esteemed director, AIP, and the MIT Institute of Pharmacy Family, welcome you all to the third day of International Conference Come Walks of AAE 2023. As this is the most awaited day of this 3-day extended international conference come workshop, the major highlights of the day being the most awaited hands-on training and workshop based on handling Autolog and Swiss ADME software to be used in CAT. May I have the privilege to welcome onto the stage the two eminent faculty coordinators of this awaited willing learning sequence from our own department, Dr. Rahul Kumar Maurya, Associate Professor Medicinal Chemistry, MIT Institute of Pharmacy, AUP, and Dr. Mohammed Yasir, Assistant Professor Grade 3, Pharmacognosy, MIT Institute of Pharmacy, AUP. I now may have the privilege to introduce these two eminent personalities from our department, those who are going to provide you intense knowledge about handling these softwares and computational tools to be used in drug discovery process. Dr. Rahul K. Maurya, presently working as Associate Professor at our MIT Institute of Pharmacy, had completed his M.Pharm in the year 2007 in medicinal chemistry from ITS Pharmaceutical College, Ghaziabad. He pursued his PhD from CSIR CDRI, Lucknow, and has been awarded with prestigious awards like GATE in 2006 and CSIR NET in 2012. Dr. Mohammed Yasir has completed his M.Pharm in Pharmacy from Dr. H.S. Gaur University, Sagar, Madhya Pradesh. He completed his PhD at NIT Bhopal, and to his research credentials, he depicts the following. He completed MPCST JRF during 2010 and 2011. He worked as an AICTE TQIF fellowship during 2011 and 2012. He qualified GATE during 2005, and he worked as a CSIR SRF during 2012 to 2016 and CSIR RA during 2016 to 2019. His H index is presently 11, and with this brief introduction of these two personalities, I would now open the forum for Sir to continue with the workshop session. Sir, please.
Good morning, students and the participants, as well as the faculty members who are from outside MIT as well as from inside MIT. So we are going to start with an introduction about the molecular docking. As you have gone through the day 1 and day 2, our immediate speakers have speak about number of alternatives, that how we can reduce the number of animals from killing or from having some difficulties. So the computer can help us to reduce this sacrificing animal. So the one of the most important tool that is usually used for preventing animal from this types of punishments, we can say punishments. So it is a molecular docking approach. So the molecular docking approach is not only help us to understand the interaction of the small molecules with the target proteins, or we can say the macromolecule. It will also give you the further steps that what you can do after molecular docking, because molecular docking is only one step that gives you the interaction of the ligand with a molecule. And if you want to prove that whether this interaction is can stand in the body, so what you have to do, you have to go for a molecular dynamics simulation. So here what we are doing, we are just try to understand that how you can first interact a ligand with a protein. Okay, in the next or in the upcoming sessions, or if the time permits, we will discuss about molecular dynamics also, that how this molecular dynamics is relevant to this alternative to animal experiments. Let me share my PPTs. So students, myself, as you know, I am Dr. Mohammed Yasir along with my colleague, Dr. Raul Moria, and we are going to discuss the one of the freeware, the molecular docking tool, which is available in the universe, that is a Autodoc tool. And this Autodoc tool, till now, you can see the number of papers, the people are working within Autodoc. So beside Autodoc, there are a number of other tools also, like they are paid. Examples are Schrodinger software and the Discovery Studio software. And this Discovery Studio and Schrodinger software, they are very costly. So the many of the institute will not able to acquire such type of a software. But if you are working with an Autodoc tool, it will give you a very good results, as well as it will give you the representative reproducible results also. So we can do molecular docking using Autodoc, and you can get the best conformer, and you can use this conformer for molecular simulation studies, that is molecular dynamics. So when we talk about Autodoc is an automated procedure that predicts the interaction of the ligand with a biomolecular target. So if we talk about biomolecular targets, it can be a protein as well as it can be a DNA also. And is as well as if you want to understand the biologics, so you can go for a protein-protein interaction also. So the docking can be done either with protein and protein. They can be done by protein and the RNA, and they can also be done with the ligand, the small molecule, as well as the receptor, that is the proteins. So here, as you know that the extracrystallographic structures nowadays, number of extracrystallographic structures were being designed, and you can see that this extracrystallographic structures or macromolecule, they are nothing, they are the proteins. And due to the advancement and the increase in the number of crystallographic structure, this will give you the basis of using this structure to carry on your molecular dynamic studies. So when we talk about a human body, and if you see the percentage of the protein in the body, it is approximately 16%, and the rest 60% is water, and then remaining percentage is of fats and other things. So what we are doing, we are considering the protein. We are considering the protein as the building block of a life. Okay, so if you consider the protein as a building block of life, then what will be the building block of the protein? So just try to understand that if you are considering anything, so that thing should be derived from something. So here in our case, the protein is the building block of the life. And if you want from where this protein comes from, these proteins are come from the amino acids. So we can say that the amino acids are usually the building blocks of proteins. Okay, so these amino acids are usually joined together with the help of the peptide bonds, and the different amino acids are joined together, and the joining of amino acids are coded by the different DNA genetic proteins, and they will give you a different structures of the protein, and that different structures of the protein will give you the action of the proteins. So here you can see this is a small amino acids. They are joined together with the help of a peptide bonds. Again, you can see this peptide bonds, or you can see these protein structures. The protein structures, they are available in the different types. So what we call them, they are either they are present in the protein primary structure. So what are primary structure? They are nothing. They are the series of amino acids. They are joined together with each other with the help of a peptide bonds. Then when we talk about the secondary structure, these the series of an amino acids that are joined together, they are started twisting, because as soon as you increase the length of an amino acid or you increase the length of anything, they will start twisting. So they can twist in the form of an alpha helix, or they can be presented in the form of the beta shape. So they are considered as a secondary structures. Now, when we talk about the tertiary structure, they are the arrangement of the arrangement. You can say the spatial or 3D arrangement of your secondary structure in the space. So that can be considered as a tertiary structure. And when you talk about the associations of the polypeptide chain, they can be considered as a secondary structure. So when we are working in the any software, or when we are working in Autodoc tool as well as Schrodinger or in Discovery Studio, there is a need of a 3D structure. Okay, so the 3D structure is very important to carry out the molecular docking studies. So here you can say in this 3D structure, there is an helix, that is an alpha helix, there is a loops, and there are the beta sheets. So they are arranged in the space. So they can be considered as a 3D structure of proteins. So when we talk about the bondings that usually occurs between the ligand and the macromolecule. Now, the macromolecule, why I call it as macromolecule? Because if we talk about the macromolecule, we can consider a protein, we can consider a DNA, also, and we can also consider any enzyme. So when we talk about enzymes, so usually these enzymes are also built up of the proteins. Okay, so we can call that all enzymes are proteins. But when we talk about proteins, so they can't work as a catalyst. So there is an assumption that all enzymes are protein, but all proteins are not enzymes. That we have to keep in mind while performing the molecular docking studies. Second thing, when we are going to interact a small molecule, when we're going to interact a small molecule with a protein or your targets. So the targets, what it can be a protein molecule, or it can be an enzyme which is present in a form of a protein. So there can be a different type of bonding which can support your ligand to interact with that particular macromolecule. So what are those? So you can see the major important bonding which can hold the interaction of a small molecule with a protein is a hydrogen bond. And this hydrogen bonding usually forms the basis of molecular docking approach. Okay, if there is a no hydrogen bonding, it gives an indication that your docking was incomplete. So beside hydrogen bonding, how your ligand can be interacted with a macromolecules. So as I told you, all enzymes are protein, and you are considering the enzymes are protein, and you are going for the inhibition studies in the proteins. So the amino acids, there is a sequence of amino acids in the proteins. Okay, so you have to take care of the active region of the amino acid. There are certain amino acids in the protein which are considered as an active site. So your ligand should interact with that particular amino acids only, so that the interaction will be complete and the inhibition of activity can be reported. So beside hydrogen bonding, you can see there are some groups hydrophobic protein atoms that enclose hydrophobic ligand atoms also. You can see there are hydrophobically enclosed hydrogen bond also that strengthens the binding of the ligand to a protein. You can also see there is a pi-pi stacking interaction between aromatic group of a ligand and a protein. So these are usually the types of bonding which strengthen the binding of a ligand with a proteins. So now we will talk about the orthodox search method. So there are certain algorithms. So when we talk about the software, so we are running it computationally, and the computational is also use some mathematical approach to find out the interaction. So usually there are certain search methods that usually take care of it. We have to take care of it. So there can be a systematic methods and there are certain stochastic methods. Such methods usually adapted, but in case of a talking studies, we are usually using the stochastic search method. So when we talk about the orthodox search method, there can be a global search algorithm. Inside the global search algorithm, you will find the simulated annealing and genetic algorithm. And in the global search algorithm, which is the solution web method is there. So what we are recommended that you should go for a hybrid global local search algorithm, and this will give you the best binding with the help of a Lamarckian genetic algorithm approach. Then there is an iterated local search, and that is a genetic algorithm with local gradient optimization. So these are usually some orthodox search methods which you find in the orthodox tool. But we will recommend you that you should go for a Lamarckian genetic algorithm because it is a hybrid of a global search algorithm as well as the local search algorithm. So the practical consideration, if we talk about practical consideration, that what problems are usually feasible when you run Autodoc and Autodoc VNA. Now, another new term comes in your lecture. What is a VNA? So it is also a platform of Autodoc, which is usually designed for high throughput training. Okay, so if you want to go for a high throughput training, you can use Autodoc VNA. What it means you can use more than one ligand simultaneously to bind with single ligand. So that is better. It is recommended that you go for a orthodox VNA, and if you want to run with one protein and one ligand, it is recommended that you should use orthodox 4.2. So what problems are visible? So depending upon the search method, as we have seen that there are a number of search methods on which the orthodox is working. Okay, so here when we talk about the problems which are visible in Autodoc, it is depending upon the search method. As I told you, we should recommend you to go for a Lamarckian genetic algorithm because it is far better than genetic algorithms, simulated NLA, and the local search. Okay, so if you run Autodoc, because Autodoc give you the freedom, Autodoc give you the freedom to use this search method. Okay, when you use orthodox simulated annealing, so when you use simulated annealing, you can only work on the problem whose dimensionality is less than 8 torsions. Means the number of rotating bonds should be less than 8. When you use orthodox with the Lamarckian genetic algorithm, the dimensionality should be 8 to 16 torsions. The torsions represent the rotatable bonds. So what are the orthodox and VNA when they are not suitable to use Autodoc VNA and Autodoc tools for performing talking. So here, when the model structure is very poor, so when you retrieve the structure, when you retrieve the structures of a macromolecule from the RNA protein data bank, you can see that there are a number of structure of a single molecule. Okay, we can say there are a number of formats of different macromolecules. So you have to select such type of a macromolecule which is an extracrystallographic structure, and the dimensions will be less than 3 angstrom. Okay, if a protein have a too many, or if a ligand have a too many torsion, more than 32 rotatable bond, such type of a ligand can also not suitable to perform docking using the Autodoc. Then if your target is flexible, if your target is too flexible, we can't handle such type of flexible target using the Autodoc. Although there is a possibility that you can go for a flexible docking in Autodoc, but if it is too flexible, if the proteins are too flexible, you can't go, and it is not recommended to use Autodoc. Now we are moving toward Autodoc, that how you will perform Autodoc. So usually the Autodoc code, it reads some file formats. Okay, some specific type of file formats like Schrodinger. It is using multiple file formats, but the master file format is .mat format. Here in Autodoc, Usually the PDB-QD format is required. So what you have to do, you have to prepare the lean PDB-QD file. You have to make the macromolecule PDB-QD file. Now the macromolecule can be flexible as well as the rigid. So in our case, what we are doing, we are using the rigid macromolecule file now. We are not going for a flexible macromolecular docking. Okay, then there is a need of order parameter file, that is a GPF file. Okay, so when we talk about the GPF file, that is a grid parameter file, it is usually depend upon the atom type. Okay, so it usually contains the Autodoc parameter file also. So when we, as I told you in the day to evening, that you have to run the Autodoc tool, that is an Autodoc suite. Okay, so when you run Autodoc suite, you will get an Autograde 4 and Autodoc 4. So you have to run to get the results. So first you have to run the Autograde 4. So when you run the Autograde 4, there is a need of the macromolecule PDB file, and there is a need of the grid parameter file, which will give you the grid maps, and it will give you the grid log files also. So when you run the grid 4, there is a need of a grid maps, as well as there is a need of a ligand and a DPF file. There is a docking parameter file, which will give you a docking log file, and this docking log file usually contains the docking results as well as the string. Oh, okay, here. So what we are doing, we will take an Autodoc platform, we will use a Lamarckian genetic algorithm, and this Autodoc will work on some scoring function. It is an empirical free energy scoring function. So when we talk about the difference between the Autodoc core tools and then the Autodoc Vina, it is the difference in the scoring function. Actually, the Autodoc is usually working on the angular force field, whereas Autodoc Pini is usually working some different scoring functions. So it is recommended that whenever you are going for a single ligand and protein docking, it's recommended that you should go for an Autodoc. So when you use the Lamarckian genetic algorithm and carrier free energy scoring function, you will get the reproducible docking results. So let us start with your workshop that is getting started with Autodoc. So what first you have to do, you have to retrieve, or you need a software first to run it. So from where this software you will get from, you have to go through this autodoc.script.edu. Here, when you write it, it will be directed to a website. Okay, that is an Autodoc website, and below logo, you can see below logo there is a resources file, and there is a downloads option. So first, what you have to do, you have to just go to the download and download according to the operating system in which you are working. If you are working on Linux, if you are working in a Mac operating system, or whether you are working on a Windows operating system. Hopefully, we all are working on a Windows operating system. So what you have to do, it is recommended that you have to download the Windows operating system. Okay, then what you have to do, when you download, it will be moved to the folder. The .exe file will be downloaded in your downloads portion. Just go to the download portion and double click on it. So when you double click on it, so what will happen? It will be get installed. It will be get installed in your C drive, and with the name of the scripts institute, the script research institute. In the program file, you will find the script research institute. Inside that script research institute, you will get the Autodoc 4.exe and autograde 4.exe file. Just copy that files for a time being. Okay, then you need a tools for working on it. Okay, now you have got the suit, and now you need the tool which work on that suits. Okay, so what you will do, you just go to the download board. So you can see the Autodoc tools are there. You just click on it and select the operating system, on which operating system you want to work. Then it will be directed to MGL tools. Okay, so this is MGL tools recommended as a molecular graphics laboratory. So it is called as an MGL tool that is usually developed by the Sangal Labs. And when you click on the download portion, it will be directed to a download MGL tools for Windows version 1.5.7. So it is recommended that you should download the latest version of the MGL tool, as I instructed you in the lesson in the WhatsApp group. So why we are downloading the latest version of MGL tool? Because if you download the latest version, a lot of bugs are there in the previous version, which are removed in the latest version. Okay, so when those who are working, those who have already worked on the Autobot, they can see that number of files are usually generated in the folders. So in 1.5, this step was removed, a number of bugs has been removed. So this is an overall flow chart that how this Autobot is going to be working. Okay, so there is a need of a key, there is a need of a lock, as we are considering our protein as a lock and the key as a ligand. Okay, so what you have to do, you have to use the autocrate as well as the autocore suite, and what you have to do, you have to run it, and you will get the different parameters file, and these parameter files can be run in the docking platform, and you can get the docking log files, and these docking log files will contain the results. So let us start the step one. Let us start with our docking. The step one is the protein preparation. Okay, so we all know that in molecular docking, there are two things that is usually required. The first is the protein, and the second one is the ligand. Okay, so why this? Why this? Okay, but if we talk about the drug discovery process or a drug design process, they can be a structure based or they can be a ligand based. So now what we are doing, we are designing on the basis of a structure based drug design. If you have a lot of ligands and you retrieve the information of the ligands from the database, and if you work on it, it is called as a ligand-based drug design. And it is usually the contiguous SAR techniques that give you the perfect prediction of it. Okay, now the first one is the protein preparation. So what you have to do, you have to get the 3D structure of the protein. As I told you that there is a need of a 3D structure in the softwares which we are in, where we are running this docking simulation, there is a need of a 3D structure. As I told you, the 3D structures are very, very necessary. So from where these 3D structures will be retrieved? So different structures, the different structures of the protein is accumulated in a certain database. So the RCSB is collecting the structures from different sources, and they are accumulating the information of the structure in their platform that is known as the protein data bank database. So we call them as an RCSB database. So what you have to do, you have to go to the rcsb.org, and when you click on it, it will open like this. Okay, so here there is a search bar. You can see the search bar in the right hand corner, and in the right hand corner, what you have to do, you just write down the protein name. So the protein which we have selected for today's workshop is a 5-IVR. Okay, the 5-IVR. So what is this 5-IVR protein? When you click on it, it will give you the information. This is the HIV protease protein. And when you look into the structural details of the HIV protein, you will find that there are two chains. There are two protein chains, and the two protein chains have the different anti-site regions. So that is why in today's workshop, what we are doing, we are taking the complete protein, and we are trying to dock a ligand which will attach to the both the chain simultaneously with the same amino acid. Okay, so people, if you have gone to the papers, you can see that what people are doing, if a protein contain number of chain A, B, C, D, they will take a single chain A or B or C or D, where the number of active sites are more, and they start off there. But in our the proteins, when they are present inside the body, they are not separated. They are in the clustering. They are in the cluster format. Okay, so the ligand should be attached in such type of conditions that both the chain should be intact, and it should be attached to a similar amino acid, which is the active side amino acid with a single ligand. Okay, so what we are doing, I think you know about the HIV protease function. So when we talk about HIV protease function, so what HIV protease is doing, as the name indicates, it is breaking down the proteins. So the proteins in the HIV protein, when they are multiplying, they are in the chain form. The HIV proteins cut down this protein into smaller fragments, and these smaller fragments are joined together, and they will give birth to a new HIV virus. So in this way, the replication process usually goes on inside the body. So So it is recommended that create a folder in your computer, whether it should be in a desktop, whether it should be in a C drive, whether it's It should be in a D drive or a H drive or H drive. Okay. And it is recommended that don't store anything in the C drive because the C drive is the brain of the computer. And if you give load on a C drive, so the function of the computer will be reduced. The running efficiency will be reduced. So for time being, what I have created is I have created a folder named Autodoc in a desktop. Okay, so when inside that, what are the important files that you are needed that the Autodoc you will perform a successful docking? First is the protein PDB file. Okay, so the first thing is the protein PDB file which you will download it from RCSB. Okay, for database. Second, there is a need of an Autodoc parameter file which is there in a .dat format, and I have shared you that file that is an atom type file that should be there in your folder because it is very much required that the parameters of the atom should be there while running the Autodoc. Then there is a need of an Autodoc 4.exe and there is a need of an auto grid for autogrid.exe. It means that you need this because you need to carry on the grid preparation and the grid log file, and there is a need of a docking autodoc4.exe. It will help you to do the docking as well as in the creation of a docking parameter file. The next thing is the ligand PDB file. So you can get the ligand PDB file from different databases, but as I told you that you can get this ligands from the PubChem database. So from the PubChem database, you just write down the name of the ligand as I instructed you yesterday. So you will get the ligand which we are using today is the rincophilin, and the rincophilin is an alkaloid. The rincophilin is an alkaloid which is usually present in the Araucaria species, and this alkaloid is recently reported to kill maximum SARS-CoV-2 viruses. So that is why we are trying that if whether this rincophilin is usually attaching to the HIV virus to the both the protein change or not. So this is our objective. So what you have to do, I think you have installed it. So we will run this exercise in live in the platform now, but just understand that what is going on. So save your energies in your laptop, plus just understand that what is going on here, then we will run. What I am doing here, you will do there in your seats. Okay. But try to understand here. So you have to go to the Windows, you have installed Autodoc 1.5.7, that is an MGL tool. And when you click on it, you will see this type of a window, this type of a window will be pop out. So this is an Autodoc tool web page. Okay. So what I told you that what is the need, what you need while performing the Autodoc, you need a protein and you need a ligand. So the two things are very, very necessary, and it should contain all the information that will be required by Autodoc to run it. Okay, so first thing, what you have to do, you have to download the protein from the protein data bank that is 5IVR. So what you have to do, you have to open that protein and you have to remove some unwanted things that are there in the protein, which will affect your interactions. Okay. So first, what you have to do, you have to go to the file and you have to open the read molecule. So when you click on read molecule, it will be asked that where your molecule is. So for today's purpose, for today's exercise, for what we have done, that we have created a folder in the desktop, and we have kept our all the necessary files as well as the protein in that particular folder. Okay. So what you will do, you just go to the file section, you read molecule. The first read molecule is your protein. So what you have to do, you have when you click on that, it will ask you where is your protein. So you have to direct your computer that please go to the desktop and open the Autodoc folder, and inside the Autodoc folder you will be able to find that 5IVR protein in the .pdb file. So when you open it, it will open like this. Okay. So it will
it will open like this. Okay. So here you can see there are certain red dots. There are certain red dots in the tree. Is it visible, students? Okay, that red dots are nothing, they are the water molecules. And if the water molecules are there in your protein, so it will hinder your small molecule that is your ligand to be interact, to get interact with the protein. Okay, so you have to keep in mind that we have to remove this unnecessary things on the protein. But there are few things which you have to keep in mind that there are certain enzymes which need two factors in the form of the metals. They should be there. Okay, that is a different concern. But now we are just talking about the water molecules and the head atoms. And when we talk about the head atom, water molecule, I think it is understandable. That is H2H molecule or H2O molecule will be there in the protein. So the next thing is the head atom. What is head atoms? So when you download the ligand from RCSP protein, and when you open that protein in a notepad, so you will find that beside amino acids, there are a number of other things are there. Okay, there is a water molecule, there is some ligand molecules are also there besides amino acids. So what we don't need other things. We need only the protein chain, which usually contain the amino acids. What we are doing, we will remove all these things. So the removal of the water and head atoms from the protein. So how you can remove it? Through water or food. So the people are using, what people are doing, people are combining the discoveries to give the water off. So it is not recommended to join these two different platforms because each and every software they work on their own algorithm. Okay, here Autotalk is working on genetic algorithm, Lamarckian genetic algorithm with Amber Forcefield. Okay, whereas Discovery Studio it is usually working on Amber as well as the Charm Forcefield. Okay, so it will give you more precise results as we compare them. But Autolog is a free tool and it is giving the reproducibility to reproducible results. That is why we are adopting the free tool. Right. So for removing the water, you just go to the edit section. You just go to the edit section. You can see the first file graphic edit. So here edit, you will find the delete water. You just select that delete water and click on it. So when you click on it, so what will happen? It will give you the warning. It will give you the warning that you really want to remove this water because it is a irreversible process. If you delete something from the protein chain, it cannot be returned back. Okay, so you have to download the fresh protein file from the database. So when you click on it, it will give you the warning and what will happen? The water molecule will be removed automatically. There are a number of other ways to remove. If the folder segment in when you look into the previous version of the Autodoc, they are usually going to give you the permission. You go to the select the function, you select the molecule or the residue or the atom from that select from the string function, and from there you have to remove the water as well as the atoms. So here, what we are doing first, we have to remove the water by using the water function of the water bubble. It is a very simple function. You just click on it, it will be the water molecule will be removed. So you can see in the structure there is no water molecule. The red dots have been disappeared. Right, students, the red dots are being disappeared. Now you have to find out whether the hand atoms are present in your protein structure or not. So what you have to do, you have to go to the select, and you have to click on select from the string, and in the string, when you click on it, so what will happen? Next. So you will find this type of a window pop up here. So this window, here you can see the atom. What I am telling you that you have to remove the head atoms. So what you will do in the head atom selection, you write down HATATM. Okay, that is a short form of head atoms. So when you write down this function, it will select, and when you click on select, what will happen? The Autodoc tool will tell you that whether or not or how many atoms are there in your protein structure. Okay, so here when you click on select, when you click on select, you can see there is some yellow color spots are shining on the display. Okay, that is an head atom. What is that head atom? This is the ligand which is previously attached to your protein. Okay, so you have to remove this for head atom because this head atom is occupying some of your amino acid which is there in the active site region, and it will hinder your interaction process. Okay, so what we will do by clicking on it, what we get, we come to know that there is a one atoms in your protein. So what you have to do, you have to remove it. So next step, what you will do, you just go to the edit portion and you have to go to the delete and you have to select delete selected atoms. So what we have selected? We have selected an head atom. Okay. So when you click on that, so what will happen? Here there will be a warning again that if you delete this, it is an irreversible process. So no need to worry. Click continue, and when you continue, the what will happen next? So this is the type of a warning. It is visible. The head atom will be removed. Now what you have done? What you have done with your protein? You have removed water and you have removed the head atoms. Okay, and this is what you are doing computationally. If you remove something, so what will happen? Then things will be vacant. Okay, the things will be vacant. So how the interaction will carry out? So depending upon your research protocol, what you have to do, you have to add hydrogen so that the hydrogen bond should be there between the ligand and the macromolecule, right? So what we have to do, we have to add hydrogens because we have removed the water and we have removed a number of things, and as well as in the crystallographic structure, the hydrogen atoms are missing. So what we will do, we will add hydrogens. So in our case, if you want to add the hydrogen, again you have to go to the edit and you have to select the add hydrogen button. Okay, so when you click on add hydrogen, so what will happen? It will give you the options. Okay, it will give you the options. It will depend upon your research protocol. When you study about the proteins, you will find out that which type of hydrogens are usually required for your study. So in our case, there is all polar and non-polar hydrogens. So what we are doing, we are just selecting the all hydrogen. So when you click on all hydrogens and when you click on OK, so what will happen? So there will be a change in the structural conformation, and you can see that the hydrogens are usually attached to the protein which was not there in the previous slides. Is it visible? The hydrogens are added. The students, you are getting my point? Yes or no? Yes, I am not able to hear you. Okay, so you have added the hydrogens. Now, what you have to do, you have to search for certain missing atoms in your protein structure. Okay, so when you look into the protein structure from the RCSB data bank, more than 70 to 80% of the proteins they usually have the missing atoms, and it is a very important step. Because if you perform molecular talking using autolog tool, number of errors usually arise because you don't know the functions. You don't know that there is need of the missing atoms, there is a need of the hydrogen bond and which type of hydrogen bonds. Okay, so here we will check whether our protein structure has a missing atom or not. Okay, so what you will do, you just go to the adding section and you have to click to check for missing atoms. So when you click on check on missing atoms, so it will give you the information whether the protein has any residue with the missing atom or not. Okay, so here what happens that it gives us an indication there is a no zero as a zero residue with missing atom. So you can type yes, okay, we can proceed further. Okay. Now, what I told you that there is a need of a charge. In the crystallographic structure, there is no charges. Okay. So what we are doing, we are trying to add the charges. So for adding the charges, again, what we have to do, you have to go to the added and you have to add the charge. Now, the different platforms, what I told you, the different software experts usually work on different algorithms and the different types of charges they are adding on it on the basis that the suitable results can be produced. Okay, in our case, in Autobot, there are two types of charges that should be paired in your coding structure. First is the column charge, and the second one is the caster charges where you are preparing the data file. So first, what we will do, we will add the column charges. Okay, so first, when you add the column charges, so the charges will add in the background. Okay, it will add in the background. It will not show you that it is adding the charges. It will calculate the charge and it will add on it. So according to this, we have total column charges added, that is 5.0. Okay, so this amount of charges is added to it. Now what we have to do, the next line, we have to add the 84 types. That is the auto talk 4 types in the coding. So just go to the added, you have to select and you have to add the just add 84 types. And if you don't do this, the error will be produced in your order of, and you will just hang up that what is going on. I am doing all the things right and right. Why this error is coming? Why the coding number are showing? And why is this problem going on? So nobody will help you out if you don't add certain type of articulating code tag on it. Now, this is the coding which we have prepared. We have removed the water, we have removed the head item. Now we have to save our coding in the PDB format. So for saving in the PDB format, so what you have to do is just go to the file and you just click on save, the save right PDB. So the when you click on here, so what will happen? It will ask you that where you want to save this PDB file. Okay, that is an important step because it will default by default it will save your file in a C drive. Okay, so if you have to give the location, then what will happen? Afterwards, what will happen? You will search where is my protein file. Okay, but I have just prepared my PDB file. Where is it? It will lost in the computer world. Okay, so you have to give the location. Computer, we can see that computer can be the universe and each and every drive is a planet. So you should know that in which planet you live, right? So here we know that we have created a folder in desktop with the name order and you have to save this file in that drive. Okay. So when you click on it, it will get save in C drive, right? Now the first step we have done, we have prepared the protein by retrieving the protein from the protein data bank. Okay. The second step now, the ligand retrieval and preparation. There are a number of ways. You can convert the ligand into the acceptable form in waterfall. Okay.
Right.
So, see, you can draw your ligand in a backend. You can draw your ligand in can sketch, okay, or different software. You can, when you draw that ligand, it will be in a 2D format. You will have to convert that ligand in a 3D format first, and you have to save that 3D format in a PDB file or a mode file because Autodoc only accepts the PDB file and the mode file, .mode file. Okay. So, what we are doing, we need a ligand. We are not applying the ligand because huge databases of ligands are there in the market. You can download this ligand from different databases. So here for our purpose, what we are doing, we are selecting the ring profiling protein, ring profiling ligand. So in ring profiling, what I told you, ring profiling is hematopoietin, which is used in the non-carrier species that is not species family, and it is recently reported to have an antiviral activity. So HIV protease is an viral protein, and we are trying to interact with the gene of protein. So it is somewhat different. So what we will do, we'll just write down the ring profiling in the PubChem. Okay, in the PubChem, you just write down in profiling, you can see that when you write down and click on it, it will be redirected into a page. So here you can see the ring profiling link is there. Click on it, you will be redirected to the ring profiling page. Okay, so in the right hand corner, click. So when you go to the right hand corner, you will find that there is a download option. So the download option will give you that in which format you want to download the ligands. Okay, you can download the ligand in a 2D format, you can download the ligand in a 3D format. So what I told you there in the media of the 3D structures when we are working on this type of the software for molecular data, molecular simulation studies. Okay. Just scroll down now, clicking the download, you will find the PD controller. So it is recommended that here you will not find the dot mole format. Okay. If you have gone through, you will find that there is no dot mole format. Okay. Usually the dot mole format is also known as the format, but it's not complete dot mole format. So what we are doing, we are downloading the ligand in the SDF format, but the 3D controller. Okay, it is very, very necessary. Now, what we have to do, we have to convert the 3D structure into the acceptable format. Okay. So, also, what format is acceptable with Microsoft? Either PDB format or in a PDB file or it may be in a mole file. And if you add a mole file and the PDB file, you have to convert them into a PDB format. Okay. So there is a software that solves this purpose that you don't go to the order of converting any living molecule into a PDB format. Okay, so what you can do, you can download the open download. Okay, it is an open download, it's an open source software, and it can convert any format to any format. Okay, you can convert any number of ligands to any number of molecules. Okay, so what we will do, I have installed this in my computer. I will show you that how you can convert this, but for learning purposes, we try to understand what we are doing. You open by clicking on Windows, the open level is open. Now, in the open level, you will see all again in the empty part. Okay, the format which you have downloaded just now from PubChem. From where you have downloaded the ligand, students? Yes, from where you have downloaded the ligand? PubChem, right? Okay, so you have downloaded the PubChem, the ligand from the PubChem in the SDL format. Okay, right? Now what you do? You go to there are two separate portions in the download. The first is the output and next one is the input. So when I stand here, this is the input and this is the output format. Okay. Now, what we have to do, we have downloaded in the SDF format. So first, tell the software that you are going to input the ligand which is in the SDF format. It will not select. So what will happen? The software will randomly select the ligand, and when you randomly select the ligand, and if you want to convert that ligand into a PDB format, it will not be converted, and you will keep the format. Okay. So what do you do? Just select the format that is an SDF format you want to do. Then go to the output format. Here in the output format, you select the PDB format. Yes, here. Okay, output format. Now output format. Below output format, you are noticing that there is an output file. What I told you, the computer is a universe, and you need to know that where you are living. In which drive you are working, right? We are working on a desktop, and in the desktop we have a folder. What you have to do, you have to give the information to the software. Please save this file in that particular folder. If you will not do that thing, what will happen? It will get lost in the computer. You will try to find out that you have converting the ligand where the converted ligand was going. Okay, so that you have to keep in mind. So just it is very simple. You insert your ligand in any format, like we have an SDF format, we will input our ligand in the SDF format and we give the location and we will just click on the converter, which is in the center. Here, the converter. And just by a single click, ligand is converted from the one component to the component. Okay, it is very easy, right? So just convert it and keep it in folder. Now, open this ligand, which is in the PDB-50 format, in your Autodoc tool. So what you will do, you will again go back to your Autodoc. Don't close any software while working because you are working simultaneously, you are working in a different software. Okay, so you are now working in a open table, you are converting to living in a different form. Now that but acceptable living format is now can be imported the code. So what you will do, you just go to the ligand. Now leave the upper bar where the file and other things are there. You just go to the ADT 4.2 here. You can see the yellow arrow. Is it visible, students? Yellow arrow. Because there you go, you can find there is a ligand. Okay, it is asking for the ligand. So you input, you just click on the ligand and it will ask you whether you have already inputted the ligand or whether you want to open that ligand. Okay, so you cannot input any ligand till now. What you have to do, you have to open that ligand. So just click on it, open, it will ask you back the ligand. So what you will do, you will just go to the desktop and you will open that Autodoc folder, and from Autodoc folder, what you will do, you will open that particular ligand. So here you can see the thing. Your ligand is open and it is in the acceptable form, no error is given, right? So for molecular talking, what we need a protein as well as we need a target. We need a small molecule. We can call them as we need a target and we need a small molecule, or we need a target, we need a ligand. So we have a target and we have the ligand, right? Now we have both the things and they are acceptable. They are converted in an acceptable, Autodoc acceptable format. Now, the first step is over. The first step is over that you have prepared your protein and you have prepared all your ligands. Now, this thing is completely different now. The second thing is the preparation of the grid parameter file in the grid box. Okay, why this? Why this? Why there is a need to create grid by just a minute. Okay, the second step now, we will do the second step. First step we have done, that is preparation of the protein and the preparation of the ligand. The next step is the preparation of the grid box, the grid parameter. So this is a very important step because you can do molecular docking by two ways. Either you go blindly. Okay, how the blind man go? He don't know the way. He can go anywhere. Okay, so that is the blind docking. You can select the complete protein and you will ask your ligand to go and bind anywhere. Okay, so we will continue. Thank you very much. So we are in the second part, that is the preparation of the data of the grid parameter file, and I told you that there are two ways that you can do the molecular docking, that you can go blindly, like a blind man go, okay, just like this is not a good. Okay, this is not a good. This is only more than the lakhs of small molecules. Okay, then only you can do it. That is more as the high explaining. Okay, so what we are doing, that we are asking the ligand to just go and bind to a particular location. So for that particular location, if you look any protein protein structure. It is three-dimensional structure of the protein. Okay, the three-dimensional structure of the protein. So you need dimensions from each and every side, as well as if you consider the cube. When you consider the cube, so here, if we are considering the cube, there are a number of blocks that are joined together. So each amino acid should be there in that particular gray box region, and you will ask your ligand just to go and bind in that particular location. Okay, so from where we can get this information that we require that particular amino acid forming? Yes. So we can get this information by different either you can go for a protein data bank, ask your website, and you can go for a ligand interaction space. You will get what are the different amino acids that are usually lies in that side region, and where you want to your small molecules point to that particular amino acid. Okay. So there are other websites also which give you information about that biological region, that is active site. There is a protein data bank Japan, that is PDBJ. So the PDBJ also gives the information that what are those proteins? What are those amino acids that you want to do the docking? There is a server which predicts the active site regions that like CASP server. So this CASP server also gives you the information that you can retrieve the amino acid which falls under the pocket regions. Okay, the pocket regions can be revealed by the use of task server. Now, here you can see that we are going to prepare the grid parameter file first. Okay. Now, before going for the preparation of the grid parameter files, what we have done, we have find out which are our amino acids that are present in both the chains of the protein, as we have selected the acylic protease protein. Acylic protease protein usually contains two chain, A chain, and B chain. And now our aim is to target our ligand in such a manner that it should interact with the same amino acid of both the chains. And both the amino acids will be in the active residues. Right? So it is a difficult task. So we are going to complete this task today. So just go to the just you go to the grid file, grid in the order of 2.2, then from where we have inputted our ligand. Okay, so in that row you will find the grid. Okay, the grid map. The grid, when you click on grid, it will ask you open here, GDL. What it means? It means that if you have prepared the grid parameter file previously, you can use that file, but we have not created any file. Okay, so we have to create it. So what we have to choose the protein first. So what we are doing, we are choosing the micromolecule first, as we have opened our micromolecule previously in our order of. So you can find it. The option of choose will be there. You just click on the choose. What will happen is window will pop up like this, where the information is there that you have one ligand and one protein. Okay, so one ligand and one protein is there. So what you have to do, you have to click on the protein by IVR. So what I told you, you can call it as a protein, you can call it as a target, you can call it as a macromolecule too. Okay, for our guys, in case of pharmacy, what we are doing, we are calling it as a target. Okay. So click on that particular portion. What will happen? Some charges which are used. What I told you, there are two types of charges usually required when you run the auto log. Okay, the covalent charges we have added previously, and here we need a gesture charges also. So when you click on it, so automatically the gesture charges will be added in the background, and it will ask you the location that says where you want to save with the charges this file. Okay, the first you have said save the changes in the PDB file. So when you click on this option, so what will happen? It will again ask you to save this file. So here, what you have to do, you have to select the protein. After selection, the new window will pop up, and you have to save the protein in the format .pdb format. Just like next, like it when I click it, it will open like I am giving the name like dot PDBQT or 5 IVR PDBQT. And our target is 5 IVR. So what I will do, I will write 5 IVR dot PDBQT and I will say this. Okay, the next step is the selection of the ligand. Okay, so what you will do, you will go to the gray box and you will select choose and you will again the window will pop out and you will select the ligand. That is the profiling. So the next step. The choose ligand. Okay, when you choose ligand will choose. Now for preparing the grid box, again, what we have done, we have selected the target as well as we have selected the ligand. Okay, now everything is okay. Now you have to create a grid box. So what you will do next, you will again go to the grid and you will select the grid box. As soon as you select the grid box, what will happen is box will appear in your screen. Click next. Here, so this red shape structure, it is a grid box. Okay, now you can you have selected different amino acids from the protein bar, and in that particular amino acids, you have created a box there. Okay, it required number of steps. So see, it's like a working in a wet lab, working in a laboratory. If you want the results, you will work 3 times, 4 times, 5 times. You will get finally you will get an exact result what you desire. Okay. Similarly, the more you are talking, it is not like you added a protein, you added a protein, and you get some particular score, and you are telling the yes, it is not like that. But you have to your every calculation depend on your parameter. Okay, your every calculations will be depend on grade parameter file, that's how the grade parameter file will be created. So here, when you click on it, the box will appear and the window will pop up. Okay, if the pop-up window, it requires certain dimensions. As I told you, it is a 3D box. Okay, the 3D box, it will require the X dimension, Y dimension, and Z dimension. What I told you, that I have selected some of the amino acids in the protein, and the dimensions I have kept 40, 40, 40. You can change according to your convenience. Okay, according to your research protocol, you can change it. Now, in the spacing category, it is kept by default 0.375. That is the intermolecular. I understand. Then the center of the grid box. So here I have made the change that you have to select the center, because when you select the dimension, the box dimension was selected. Now where this box should be kept? Okay, first you have selected, first try to understand that you have created a box. First you have created a box with XYZ dimension. Now a box, where should this box should be there? आपने box बनाया, वो box कहाँ पे रहेगा? Okay, that is your A. So that is how you have selected your amino acid. So once you have selected the amino acid, you will scroll down, right, green and blue. When you scroll down this, your box will move up and down and reach in every direction. So just take this, box and keep it in that particular in that particular location. Now what you can do, you can rotate. As this is a cube structure, so you are working at 3-dimensional. So you should know if you work like this, you will take a box and if you keep anywhere, it's not necessary that other dimensions are covered. Okay, it is not necessary that other dimensions are covered. So what you will do, you just rotate your 3D complete protein. When you rotate your complete protein in the help of a mouse, it will give you the information whether the protein, whether the amino acids are completely covered or not. And it is a very important step. If you will not cover this step, so what will happen? Your grid parameter file will not be created up to the mark. It will create, but it will not up to the mark as you required. Okay, then again the results will suffer, right? So after selecting the dimensions as well as after setting up the centers for the grid box, what we will do, we will save this information. What you have to do, you can see here there is a filing section was there and the close saving current was option was there. So just go to the file and close saving current. So when you close it, it will ask if the information will be stored. Now what you have to do, you have to create a file. If you have to create a file, see, in other softwares like this Studio, Apple Studio, the files are automatically created. Here you have to do some work on it. Okay. This is a free software and a very good software, so you have to do some work. So first you have to go to the file and you have to select on close saving torrent. Then what you have to do, you have to put the grid, and then you have to select the output form, and you have to select the output form, and you have to save the file in the grid as a GPF format, grid parameter file format. So when you click on it, the video will pop up. Now again, it will ask you, "I kahan save karna hai?" So what you will do, you will go to the desktop where you have created a folder, and you have to save that file in that particular folder, and you can check, you just click on it, you open that GPF file in the notepad, and in the notepad you will have all the information that what parameters you have selected, what dimensions you have selected for creating a grid parameter file. So this is how you will create a grid parameter file. You have to check it by opening the Autodoc folder. So you have to check it out. If it is not there, it means that it is not created, or it is lost from your errors. Okay, so what you have to do it again, you have to cross check whether the files are created in that particular folder or not. So it is very, very important that you have to keep in mind that cross checking that whatever you are doing, it should be there in your folder. Next. Now, we have a grid parameter file, we have the protein, we have a ligand PDB file. Now we have to learn the auto grid. It will give you the information that whether you have done each and every step properly, so that the different maps have been created, electrostatic potential, desolidation energy, all should be calculated by using this auto grid. So what you have to do for running the auto grid, so you just put that Autodoc 4.2 lane, you can see that there is a run option for them. So click on run. When you click on run, there are two options that you will. The first option is whether you want to run auto grid or whether you want to run the Autodoc. Okay, so we have not done anything to run the Autodoc. Okay. We are in the third step that we have to run. We have to run the auto grid so that our grid, the log file should be generated. So what you have to do, you just go to the grid thing, and you just go to the run and you click on the run auto grid. Okay, so when you click on it, the window will pop up. Okay, like this, the window will pop up. And here, the important step you have to keep in mind that we have downloaded the Autodoc, which is Autodoc 2. Okay, in Autodoc 2, you got Autogrid and Autodoc 4.exe file. So what we have done, we have copied that Autodoc 4 and the Autogrid 4.exe file, and we have kept that file in the Autodoc folder which we have created in the desktop. Right? So then what you have to do in the program path name, so you have to give the program path name. If you do not select the correct program path name, your grid auto grid will not be able to run, and the error will be produced, and you will see that number of errors will be there, and this error will give you a trouble, and you will after some time you will get panic and you will shut down the computer. It is not possible. So this is not the attitude of the researcher. You have to find out the problem that what is going on, and you have to solve that problem, and you have to try to solve that problem at least. So what we will do? We will go to the program path. So program path, what is the program path? It is there in the desktop, right? It is there in the desktop. There in the part of it, not a code.exe file. So what you will do? You just browse it, and you just go to the Autodoc 4 and you click on Autogrid 4.exe. Right. So when you click, the correct program path name will be selected. So once it is selected, the second option is you have to select the correct file name. So what file name we have created? Yes, students. What file name we have created this from? Participants, we have created a doc.gpl file. Right? We have created a grid parameter file right or wrong. Okay, what name we have given? We have given the name doc.gpl and we have stored it in our desktop version. Okay, that is your file name. So what you have to do, you have to select the correct file name first, and when you click on it, you just the login for the grid log file will be automatically generated, and you just click on launch. So when you click on launch, what will happen? Depending upon the processor speed, okay, depending upon the computer processor speed, that it will take some time to run. Okay, as I am running in i7, you will run in some i3, some of you will run in i5 processor. So when you run, the speed will be different. Okay, so it will it will take some time to generate the grid parameter. Well, so as if you run in a high computer or if you run in a high-end system, so what will take it will be done like this. Okay, and if you run like run in a Linux platform, it will also be running in 2 seconds. Okay, because time taken in the Linux system is about 2 as compared to the Windows. Now, we will first look whether our grid log files are being created or not. What you will do, you just go to the order of folder which is created in the desktop. Okay, just click on it, double click on it. You can check it out whether the grid log file is being created or not, and whether it is being successfully completed or not. Okay, in the previous version, what is going on? Some people are using the previous version like 1.5, 1.5.6. So if you look in that previous version, all the files will be there in front of you. There is no need to cross check by opening the files. Okay, in 1.5.7, you will get a single GPL file with some size. Okay, so here our doc.glb, that is the log file which is created by running the auto.exe. So you will see the file size is only 1 GB. And going forward on the lower processor, just refresh it. You can find out whether it is being created or not, or the size of the file is being increasing or not. Okay, so what we have to do is check out whether the grid log files are being successfully run and all the dimensions are being created or not. So what you have to do, you have to just click on right click on it and you have to open that file in a notepad. Okay, because when we go and open it in that in a notepad, so what will happen? The notepad will open all the data. Okay, it will give you all the log information which is done by the auto grid for.exe. So here, when you move down, so you will find that there are a number of maps are created, and these maps are necessary. These maps are very necessary to calculate the document fractions. Okay. So, here, while in the lower side, you will find that there is a successful completion. Okay, there was written successful completion. When you do run AutoGrid 4.exe in auto 1.5.7. So what you are doing is to cross check it whether it is a run or it is not. Okay, it is very, very necessary that each and every step should be run successfully. So here we got an information. Here we have got an information that it is successfully run. It will give you the time, how much time the computer has taken to run the grid parameter file, and it will give you the grid log file. Okay. So next. So this are the information which you can when you read the grid log file, there are certain codes that are written like HSTSC, HSTAMNAS, WALSF. So these information you can get from this slide. I will share with you. Now our fourth step. Okay, the fourth step is the preparation of the topic parameter file. It is the dot .dbl file. It is not as .dbl file. So till now, Quick recap, what we have done, we have created a protein PDB file, we have created a ligand PDB file, and we have created a broad grid parameter file. We have run grid or grid, we have got the grid log file. Okay, now what we have to do, we have to prepare the docking parameter. And again, what you have to do, you just go to the area, you will find docking, blocking. Inside the docking, what you will have to do, you have to select the macromolecule. Again, when you click to select the macromolecule, it will ask you in which format you are working in a rigid following or you are working in a flexible. Again, what we are doing, we are working in a rigid filing. So rigid protein structure. So we will select the rigid protein structure and we will click on it next. So when you click on it, your protein will be selected, that is defined by here. Now there is a selection related to the same, you will go to the topic and you will select the ligand. Now you will not, now there is everything is there in your videos. So what you will do, you will just do, when you click on it, what will happen, the ligand profiling will be selected. So this is the form, and you have to select the in the file. Next. Now, as soon as you will select the ligand, what will happen? The orthodox ligand parameter file will be used. So it is recommended that you use the default parameter until and unless you have not gone to an extensive literature survey of your ligand as well as protein. Okay, so what we are doing here, we have chosen everything, ligand item tags, number, everything, and we have selected, and it is an range, and we will accept the default parameters. Now, the second step is the search map parameters. As I told you, the search method which we have discussed in the previous slides, like orthodox usually work on a different search algorithm, like they can work in simultaneous learning, they can work in a genetic algorithm, they can work on Lamarckian genetic algorithm. So what we are selecting that we have to go for a Lamarckian genetic algorithm. So we will go to the search parameter and we will select genetic algorithm. As soon as you will select genetic algorithm, the genetic algorithm parameters will be open. Now, depending upon that, this parameter that is very, very important for the talking purpose. See, if you go through the research, if you go through your research and if you are making a protein structure, by how does you normally or you have just prepare a protein with the help of a modeling approach. So you should know about certain parameters of the protein and how you are doing log such that the protein with the help of this genetic algorithm parameters. So you can adjust this parameter depending upon your research protocol. And it is recommended, all the values are set universally and it is recommended to select the default parameter. Don't try to change until and unless you know everything about it today. Next. Now, you will have again what you have to do, we have to go to the top parameters and we have to first what we have done, we have adjusted the search parameter. Now we will adjust the top parameter. Similarly, what we are doing, we are selecting the default parameters. Next, so when you select the top parameters, the new window will pop out. And here you can see there number of options are there. The seed libraries, the set, the set fields, customize energy parameter, customize step size parameters, and customize output parameters. So the customization can only be done if you will provide. So it is recommended don't change any option. Accept the default parameter because they are set to their minimum range and they are universally accepted. Now, accept the default talking run options. Now, what you have to do, you have to add the default parameter. Just go to the docking and you can click on other options and you can select orthodox 4.2 parameters. All the orthodox parameters will be added. You just click on and select next one. Accept. Now the output option. Okay. So now what what we have done in the gray cabinet, while we are creating a gray cabinet file, we are receiving the gray cabinet file, and we are using this gray cabinet file in our Autodoc, auto-before.exe to give the output which is in the gray block file. Similarly, we have created a document parameter file. Similarly, what you have to do, you have to output and you have to select genetic algorithm. Okay. Here. So here you can see there are number of other search output options are there. You can select it. What I told you, the European genetic algorithm is far better than simulated analogy. Okay, so it is always recommended to choose the American genetic algorithm for blocking countries. Next. So what you will do when you select the American genetic algorithm, it will move to a window. It will ask you to save this file. So what you will do, similar, you give your DI name, 1, 2, 3, 4, 5, 6, whatever you want to do, you can write any name on it. Okay, it hardly doesn't matter, but you should know that what name you are giving to that particular file. Okay, the here what I have done, I have again given the name dot DBL stand for dot file, and you can save it. Okay, you can save, I can cross check it, go to the Autodoc folder in your desktop, and you just take it out, whether your document file has been created or not. Okay, yes, it is there, and it's created. Now the second step is the running of the Autodoc, right? So now we have done the running of Autodoc. We are in the last step is the running of Autodoc, and then we will be in the video step that is analysis of the result. First, you just go to the run option again, run inside the run, you select run Autodoc. When you click on run Autodoc, first what you have done, you have run Autodoc. Now what you are doing, you are selecting the run Autodoc option. Next, so when you click on it, so what will happen? It will ask you give the program name, give the path name, give the file name. Okay, so it's need not to repeat. You should know what parameter file you have created. Students, what is the name of parameter file? Dot parameter file. That's so you tell them it is a dot parameter file. That is your parameter file. Now, what is the program path name? You have to select it. So, like, हमने कहाँ पे रखा हुआ है अपना Autodoc code.exe? It is in the desktop. It is under the Autodoc folder. Now, select on it, and you just click on the launch. Launch. When you click on launch, depending upon the speed of your system. Okay, depending upon your speed of your server, it will take time. So if this type of a window will occur, it will give you the information, yes, the program is running properly, and there is no any bug or any situation that is to be handled here. So after some time, what will happen? If this window will be disappear, it means that the Autodoc is being completed. Now, is it completed? Okay, so what you have to do, you have to cross check it. For cross checking, what you have to do, again, you have to go to that window, you have to click on the talking.dlg file. First, you have to select that whether the DLG file will be created or not. If it is not created, it means that there is some problem, there is some mistake, there is an animated errors. Okay, then again, if it is created, you have to say, is it successful or unsuccessful? Again, same what you have to do, you have to right click on it, you have to click on the notepad, and you have to open the .dlg file in the notepad. Okay, so when you click on it and then you scroll down, there is a successful completion, and it will tell you that how much time has taken to perform your Autodoc. Okay, so we have done the successful completion of document now, and it gives you it gives you the information that yes, your Autodoc.exe has been properly learned without problem. Now, the important step is our objective is being achieved or not. What is our objective? Anyone of you? Please provide them. Anyone of you, what is our objective? Anyone? Whatever you want to say, according to you. Any one of you? Please, remind me.
So to get the binding energy, what we require, what we compare with what we want the bind energy like standard if binding energy more higher with the standard then we will get
Okay, okay, it's okay. Please transfer the mic to the. You can see this, so we want to stand up.
If you binding energy to identify the binding energy or binding interaction, ligand interaction, or conventional hydrogen bond.
Okay, okay. Anyone else?
check the compatibility between the ligand and the binding.
Okay, anyone else? I think someone has raised their hand. Okay, easy. Sir, as my friend said that we have to see the binding energy. So, because we are not creating the molecule in the wet laboratory. We are just visualizing before doing actually in the laboratory. So we have to see the binding energy and we have to see how compatible the ligand is to the protein. One last try. Anyone wants to try more? What is our objective? Anyone wants to try? It's this is not a classroom. It is about
Make these two together to all and
see, I told you that while you were in the molecular talking, you get the binding. This is not our objective. Our objective is whether our ligand is binded to both the chain with the same amino acids in the particular region. This is our objective. Okay. Binding energy, we will get the binding energy. If you do the docking, you will get the binding energy. You will get the binding energy, will have the less binding energy, some will get more binding energy. But what I told you, what is our objective? We have taken two chain, we have taken a child protein, and child protein is connected to chain, A and B. Okay, and A and B both contain the active sites region. And our objective is our ligands to bind in such a manner that it should bind on the same amino acid in both the pockets, both the chain, both the proteins, right? So now we will analyze our result and we will find out whether it is being achieved or not. What you have to do when you run your autocom and you get your successful completion information from the system that your docking log file is successfully completed. So what we have to do, we have to open that log file and see if it will give you the information whether it is done or not. Okay, so what you will do, you will just go to the analyze solution. We will go to the analyze, click on open. So what you want to open? You want to open the log file. Okay, so which log file you want to open the log file? So just click on open, where it will ask you where is your log file, and just select the particular folder, and you just reach it to the particular folder and click on the DLG file, and when you click on that DLG file, what is the file name? Yes, anyone will be. What is the file name? Dog.dlg, yes, the dog.dlg. So you want you have to open the dog.dlg file format. So just click on it and you the dog.dlg file will open. It will ask you that please. It will ask you that yeah, do you want to give a new name? Yes, give the permission because it is not issue to us. The issue is whether our objective is being obtained or not, right? So we'll give okay, yes, okay. Then it will give you information here. Then dog confirmations are being achieved. So this then dog confirmation will give you the information whether your objective is being achieved or not. So you will click OK, then you will go to the analyze third. Again, you will go to the docking. The docking, what you will do, you will have to choose the macromolecule first. what kind of molecule, when you click on it, window will be pop up and it will give you this, it will give the information that you have to select the high value given protein. So when you select on it, it will the screen will completely change out. Abhi the blackness on the red will be converted to white. Okay, the white screen. So you can change the screen. This is part of matters. Okay, but naturally what is going on in order of we are doing here. So it will give you information here. There is a light inside interaction display option. So this will give you the information that if you want to change the display option, you can use this parameters here that you want your molecular surfaces to be displayed. If you want your close contact vectors to be displayed, if you want your hydrogen bond to be displayed, so you can click on it. So what we will do, we keep it as it is, and we will go to again docking, and we will ask, we will go to the confirmation, and we will ask with plain confirmation by energy. Okay, so it will give you the result of the confirmation first, which have the highest negative energy. Okay, as in the molecular we are working with negative energy. If the energy is in the negative, it means that the binding state. There is the positive energy, we will not consider such type of resolution or problem. Here, analyze information and play line by energy, play to play line by energy. When you click on it, so what will happen? Let's see. So here, this complete picture will be displayed to you. Okay, so I will go there, I will go here and there. First thing, see, as soon as you will open it, what will happen? The first thing that will be displayed like this. Here, this is Ringo file. Okay, please take the cursor. Okay, the Ringo file. So this is the navigation part. This is the navigation part. You can what you can do that you can click to the next information by using this navigation part. But this navigation part is far more than this changing of the confirmation. If you want the information, what is the binding energy, what whether the bonding is taking place, and other information, you can ask it later. So what you have to do, you just click on the and window. So when you click on the and windows, and ends. Yes, yes, this one. When you click on it, so what will happen? This information will be appear. So the set play options will be there. Sorry, the set play option will be there. In the set play option, first thing what you have to do, you have to click on show information that is written as a show info. So when you click on show info, it will give you information what is the minimum binding energy which you have. G. And the second thing, what you have to do, you have to find out the hydrogen bonds with the ligand and the protein. So it will also give you the information that whether the hydrogen bonding has been taking place or not. Okay. So here you can see in the confirmation interface. So in the bottom there is 5 IDR A gene and the 5 IDR B gene, right? Is it visible, students? I'm not able to hear this one. Okay, so here you can see aging and aging. Now it is isolation 50. Okay, so isolation 50 is our amino acid which falls under active site residue. Okay, what is there in there? 5-IPRB chain. Which amino acid is there? Yes, students. Is it visible? Is telling me in B chain, which amino acid is there? Anyone of you can use minds.
Isoleucine.
There are a number of isolations.
50.
Yes. Okay, the same residue as this can be in the A chain. Okay, in A chain, the 5 area, HIV proteins, A chain, isolation 50, and HIV, protease, pyruvate, EJ, isolation 50, both lies in the active site residues. Okay, and our ribofylline binds to this both genes. Okay, now what we do in the wet lab, we do practicals number of times. Okay, so that we can give information in a statistical manner. Okay, so you can give any mean as well as you can assess the result if it is reproducible by number of three, or if you repeat the experiments by three or number of times five. You repeat such type of experiments, you don't put the readings, and you can consider that here this is our right results. So finding this, what you have to do, you have to do for the clustering information. Here, the binding energy with the top of the conformation board is minus 5.7, 5.27. Okay, so what you have to do, you have to find out. Okay, you have to find out that how many times this conformation has taken place. The conformation means the binding of the group, binding of the ligands to the protein, it should be more 3 times. Okay, it is only acceptable. Okay, so the people are what they are doing, they protein to drop it and get the result, this is good. It is wrong. Okay, this is completely wrong. So what you have to do, you have to repeat the experiments number of times so that you can get a proper answer, as well as you have to see that your conformations are in the same protein amino acid residues. Okay, and it should be important to that. So what you have to do is for check it, you have to go to the analyze portion, and you have to go to the clustering, and you have to click on show clustering. So when you click on show clustering, what will happen next? It will give you the clustering information. Wherever you have to do analyze, you have to click on clustering, and you have to click on show cluster. So just when you click on show cluster, what will happen next? A bar icon will be appear. Here is the binding energy, and there is the confirmation. The confirmation is the same amino acid of chain A and chain AB. You can find that 6 confirmation has a single docking score. 6 confirmation out of 10, 6 confirmation have the same docking score, and whereas you can find the 3 confirmation have the same docking score, and 1 will have it. So we will not consider 1 and this 2, 3. Okay, other if what happens if this 1 is there in that place, the high standing energy will not consider it. We will review the standards, repeat our documents that we will change our requirement profile, and we will again find out the perfect score and the perfect. Well, thank you all. That is all about the order of it, how we want to run. Now we will run this in a like, okay, we'll open the order, we'll retrieve the files, and we'll run in the Thanks. So I am Dr. Yasser, and my colleague Dr. Rao Srivastav. You can reach us. Our phone numbers and email address are there. If you have any doubt or you have any queries, you can reach us out on my phone number as well as my email address. So are you ready, student? You can start playing the order of your platform. Yes or no?
Please switch on this line. Yeah. You are late. Not all lights. Seriously.
Open other than this light.
And give me the first one. Something. जो भी नहीं करेंगे डुप्लीकेट बनाएंगे वो हो गया आपके पास बस आगे Okay, students. So let us start exercise. So you will see that I have created a number of students in the desktop. So what you have to do, you just click on that auto lock. Okay. So when you click on it, so you will find the information that what we have to keep. Okay. So you need a file idea of 14. You need a file idea of 14 and you format. Okay, just now I told you everything that you need a format, you need a format, you need autoclick and mother oxford.c. Okay, so these files are very, very important. And the one thing that is the 84.1 underscore bound dot that file. So that is also very important for it. So these particular files are necessary to run the operations. So we will start the order of software and we will try to done just now what we have to do. So I am deleting the previous information here. Okay, you go, you will
Students, so you have to go to the windows, you have to go to the windows and you have to type auto go to windows and you just type auto 1.5 you click open. So when you open it, what will happen? Click on open. So it will take a few minutes, so it will be launched. Okay, so this command center below activate and it will give you the information that or open to find is invoked. So you will just wait for a few minutes and you will get an information and it was opening. It will give you the option if you want to register or you want to remind you later. So what you do, the remind you later, the version will be turned into the red color. You have to wait for some time until the auto has been loaded to the person. So here you can see loading module. Okay, loading module where it is 7% now. It is taking time, depending upon the processor, it will take some more time. In my right case, it will take hardly 30 to 40 seconds. Here it is loading it and it will be 100%. You can see there is a green button register now which is activated and there is a remind later button which will be activated after only the 100% loading of the module. So please be patient and you load it until auto will be loaded. Here, after 100% loading the module, what you have to do, you have to click on remind me later. Just click on remind me later. So your auto module will be opened. So what you do is just enlarge your screen. Okay. Now, what I have to do, you have to open your coding. Okay. The coding you have downloaded from the RCS coding data bank. So here, what we'll do, we'll go to file, we'll go to the molecule. Student is visible to you. Okay, read molecule. Now it will ask you where is your molecule. So what you have to do, you have to go to the desktop. You have to go to the desktop, and here this desktop which usually contains the autodoc. Okay, this is your autodoc folder. You have to move to that folder, and here you can find that here is a 5 coding in the PDB format, 5 coding in the PDB format. You can see I done not here, but what I will do, I will select the 5 coding in a PDB format. So when you click on it, and you just open it, you just go and open this. So what will happen? Your coding will be open in the display portion. Okay, so here in the display portion, you can see there is a red spot and nothing. These are the bottom of the queue. So you can zoom, you can zoom out, you can recall this protein. Okay, you can rotate this protein according to your convenience. Okay, so here you can see there are a number of options are there. Whether you want to select the protein, whether you want to see the protein in the sticks and the ball format, whether you want to display in the atomic sphere format, whether you want to display in the ribbon format. So ribbon format, everyone is interested in the ribbon format, but if you are really a chemist or if you are really a biological bioinformatics, so what you have to do, you have to go for a you have to go for a sticks method. So you can click on it. So what you want to do is see the protein again on the coordinate instruction format. Okay. So what we have to do, the first thing we have to remove the water and we have to remove the atoms. So what we are going to do, we will remove this. Okay, because it will make us make trouble because the ribbon will cover most of the portion. So just go to the added portion. You will find the delete option is there. Delete water. If you click on delete water, all the red spots will be deleted. Okay, the water water molecule is being deleted from your protein. Now what you have to do, you have to remove the hat atom. So what you are going to do, you have to go to the select portion and you have to go to the select from string. So when you click on select from string, so what will happen? See this type of window will appear. It will ask you that what string you want to delete. So what I am doing, I am writing the atom HETATM. Okay, hat atoms. So when you write the hat atoms and when you click on add button, so you will find that an atom is being denoted by a yellow color. So you can zoom out. You can zoom out. Here you can click on the floor. You can zoom out. So this is our head atom. Is it visible, students? Okay. So this is our head atom. So this head atom is really occupying our minds, which are necessary for our DNA to bind a particular protein. So what we will do, we will remove this. So just select on it and click on dismiss. Go to the added, delete selected atoms. So when you click on it, it will give you the warning. It will give you the warning whether you want to delete it or not. Okay, so what you have to do, you just click on continue here. So your added in will be on the screen. Is it okay? Students still now? Okay, so what we have to do now, the next step is students, participants, we have deleted our water molecule, we have deleted our hydrogens. Now what you have to do? We have to add hydrogens, right? So what we have to do, we have to go in the added portion and we have to click on hydrogens and add. So when you click on it, it will give you the information that which type of hydrogen you want to add. So I told you that depending on your research, you can select what type of hydrogens you want to add. If you want to add only polar hydrogens, you want to add non-polar hydrogens also. So we are, in our case, we are selecting the all hydrogens. So click on OK. The appearance of the protein will change because the hydrogen atoms are being attached. So you can see these are the hydrogens which are white, which are appearing in the white, and we just use the attached. Then next step, what we have to do? Yes. Now we have to add the charges in the atom types. So what we have to do, we have to go to the added, we have to go to the charges, and we have to add column charges. Okay, before adding the column charges, what I told you that you have to find out whether there is any basic recipe or not. Okay, so the first, before adding the charges, you have to go for a missing residue. So what you have to do, you have to go to the miscellaneous and you have to check for any missing atoms. So when you click on it, it will give you the information that zero residue is missing atoms, right? So click OK, yes, our protein is okay, there is no problem with our protein. Now we are going to add charges. So you just go to the select portion, added portion, you go to the charges and add column charge. When you click on column and charges, the charge, this is 5.01, as I told you in the slides, it's being added on the proteins. So what you have to do, you have to click OK, and you have to add atom types. So you just go to the added atoms and assign AD4 orthodox 4 types. So just click on it. Your orthodox 4 atom types will be added. It will not be displayed, but background it will be added. Now the second last step is the saving of this protein in a PDB format. So what you have to do, you have to go to the file, you have to go to the save option, you have to go write PDBQT. So when you click on it, it will ask you, see, the file name, is it right? Participants, the file name, is it right? Yes. How many pages raise your hand? Yeah, raise your hand. We want to raise it. It's not a classroom. Okay. So it is wrong. So it is wrong. Many of you are individual. So what I told you, we have kept all the information private. Now my side information. We have our information are there in the desktop. Okay, where we have created a folder name or log. So you can change that thing. It is very important. It is very important. If you forget this thing, your protein will be lost. Okay, you will not be able to complete your time by using the order log. So browse then desktop. You go to your folder, which is there in your desktop, that is an Autodoc 4, and you can click on the 5IBR option and you can save it. It will ask replace because it is already created, so I'm replacing it. But now which protein I have created, I will add this on it. Then, okay, so your protein is there in the PDB format in the Autodoc folder, there in the text. Then the next step is our protein. Our protein target is here in our screen. Now our second step is the input of the ligand. So what you will do, you will go to the ligand. You have to open your ligand. Our ligand is there in our Autodoc folder. That name of that ligand is the ribofylline. So we have opened our ribofylline protein, and it will give you the information about the TORS dot profile. It means that the dimensionality of the protein and the number of rotating bound in your ligand and the dimensionality of the ligand is a 5, which is acceptable, and you can perform this docking of this ligand using the order. You click OK. So here you can see our ligand is. So we have only one screen. Okay, we have only one screen. So where the computer will show you that you are ligand, it will be in the same screen where your molecule is. So what you can do, if you want to look your ligand, you click on it here. You just click on this option in front of IVR. In the front of IVR last, you can see the red spot. You just click on display and hide. Okay, so here you can see this is our ligand, which is this is the ribofylline, which is given the 3D format. So from where we have downloaded the ribofylline from Pakistan in the 3D format and STL format. So we have to convert this STL format into a more format. So I will open now the Open Babel just to introduce you how Open Babel is usually working. So just go to the windows, no need to close your Autodoc. You just write down Open Babel. Okay, here it is there. You just open it. This is Open Babel GUI, graphic user interface. It is opening. So we will give you. Is taking minutes to so here the fish is the logo of Open Babel. So this is our Open Babel GUI, the graphic user interface. So here you can see the output format in my backside. You can see the output format. I think it is being hidden by Zoom. Okay, now it is visible. It is the output format, the lower than the input format. Okay, so here when you click on the input format, just look the types of ligand format which can be converted with the help of a open label. Okay, so here you can see approximately 100 to 200 formats of one ligand can be converted into a other formats. So what we will do, we'll just select the SDF format. This SDF format because we have downloaded the thing in the SDF format. Okay. Click on SDF format. So you will see that in the imminent format, your SDF mode format is selected. Now you in the output format, you can see it is an orthodox PDB format is there. You just go to these three lines. The three lines where my arrow. Please look it up on the screen where my arrow is moving. So in this three dots, you just click on it. Okay, when you click on it, it will ask you where is your ligand in the SDF format. So what you will do, you just go to the desktop, you open the Autodoc and you will find here. It is not there now, but you will find that ligand. If you will store that SDF info file format, it will be there here. You just click on it, it will be get inserted. And the most important thing is the output format that you have to select the output location that way you have to save that converted ligand because only the PDB file format is accepted in our AD photo document. Okay, so I will close this. If the time permits, we will have conversion of a ligand from one format to another format with the output and open it. But for now, I'm closing it. So because we are reaching to the lunch time. It is 1 p.m. So our ligand is there and our protein is there. Now what we have to do is students. We have our protein, we have our ligand in our screen. Now what we have to do? Students, please provide the mic to the participants. Yes. Yes, the next step is the generation of the grade parameter file. Okay, so what we will do, we will just go to the Autodoc 4.2. That brown color line, you should see that brown color line is there where the AD. Yes. So can you please tell after putting the input, how will you convert it in the output? Because okay. So you have your open web with you. Yes, you have installed open web. Yes, sir. I've inserted the input in the view format, okay. And I've selected the PPT to be output. You have selected the location. So the location of input. Yes, anyone of you can help me. Please look at his laptop. In the output portion, you will have three dots. Three dots are there. Three dots. Yes, sir. Click on three dots. Yes, sir. In that dot, it will be directing you where you want to save. So what it is reflecting. Yes, sir. The location from here. The location will be where you have kept all the files. Okay. So select that. Yes, sir. So select that location, select that location, write down the name of the ligand profile. Okay, then click on convert. Just click on convert. It's your click the convert button. Yes, now it's okay. No need to save. It is saved automatically. You will go to your folder. You will find that PPT file is there in your folder. Is it? Yes. Thank you. So now we will try to detail of a great parameter file and we will try it and model it. Okay, so what you have to do is told you that go to the grid option, select macromolecule, and you have to select choose. So when you click on choose, what will happen? It will give you it should be clear in your mind if you are writing the name of the protein or you are saving the name of the protein with other name, it should be clear to you. Okay, so what I have done, I have not changed the name of the protein. The name of the protein is itself the file idea. So what I will do, I will just select the file idea and click OK. Like this, you have to select and then select the molecule. Okay, so the gesture charges will be calculated and it will be added on it. So when you click on OK, some information will be there. You have to again save your ligand in a liganding format because in the first step, we have added the polymer charges and we have saved it in a format. When you open the same protein in a AutoCAD option, you will add the gesture charges. Now that both the charges are added, now you have to again save the protein in a BDPD format. So where we are saving? We are saving in a desktop. So this is our 5 IBR ADP format. You have to save it. It will ask you whether you want to replace. You have to replace it. Okay. So this we have selected our protein. Now the second step is go to the grid. Maybe we will be little bit faster because I have taken much time. My colleague is waiting for me so that I should I should cover my section as soon as possible. So grid, you will select map type, select map type for आप किसके लिए डॉकिंग कर रहे हो आप कर रहे हो अपने particular ligand के लिए. So what you will do, you have to choose your ligand which is already open in your Autodoc. You have to select the input filing and you have to select the ligand. Okay, just so again, what you have to do, you have to go to the grid, you have to go to the grid box. So when you click on it, the same grid box will be created, right? Okay, dimensions there. Yes, students, anyone remember the dimensions? किसी को dimensions याद है? Yes, we have to adjust this. The centers, the grid box centers as well as the dimensions. X, Y, Z dimension. What is it? Anyone of you? Even I don't remember because I have done it. I think it is 40, watts, right? It is the X, Y, Z dimension 40, 40. What is the dimension of the grid box? Anyone remember? So I am giving you the dimension, you write it down and tell me. Okay, just a minute. I am opening my PPTs again. Okay, write down the dimensions. Okay, students, very this is the law. So, okay, any one of you, please write down the X center. The X center minus 12.95 minus 12.95. The Y center 21.16 and the Z center that is minus 5.4. Okay, we just write down. I will ask my colleague Dr. Rahul to just go to the and just set this sentence. Close the PPT. You don't need any PPT. Here, what we will try that the dimension x, y, z dimensions are 40, 40. Now, what we are doing, we are just adjust the center x, y, z center. So please write down. Anyone who tell me what is the x center? Minus 1 minus 12.985. The y center. 21.116. Is it? Let's mark that. Okay, the y, now y center minus 5.42. So now what we are doing, we are just rotating our red box and we are trying to just look that all the minus usually fall under that particular box or not. So here you can click rotate like this by using the mobile and by using the mouse. You can change the view also, like you just don't want to look it in the box format. You want to look into the line format, so you can change the display. So you can see the line box there. The line box that is in the green and the red color. Okay, so it is very hard to understand where is your gray box is. So what we will do, we will remove this box. Okay, now what we are going to do, we are going to save this. We will go to the file and we will choose close saving current. Okay, so once it is being saved, now what you have to do, you have to go to the grid and you have to go to the output and you have to save the GPM. So what I will do, I will save the grid parameter file in the same folder which is there in the desktop. Okay, so this is the doc.gbm file. It is already there, so I will create it again. We will save it and we will replace with the current. Okay, so we'll check it out. We'll check it again whether our grid parameter file was there in our folder or not. So what we will do, we'll again go to the folder, we'll go to the desktop and we'll go to the Autodoc folder and we will find out whether our GPF file is there or not. So this is the GPF file which is usually there in our, so we will click on it. So you can see the date of, you can see the dates are being changing because we are changing, we are replacing the things which are already done. Okay, the change is the 3rd November 2023. So you open this, right click on it, you open with Notepad. So when you open this with a Notepad, you will get the information. Okay, so this is the information we have had. The X, Y, Z dimensions are 40, 40. Okay, the third, the grid center, minus 12.985, then 20.16, and then minus 5.42. Okay, so this is the entry we have given to us. Okay. Then we'll close. Okay. Our grid parameters files are according to what we said in order of right. So I have deleted the previous grid log file. Okay, now I am going to run the auto grid. So what you have to do, you have to go to run auto grid. Then you have to select the program path name. Again, is it right program path name? Yes, students. Yes or no? How many of you say no? How many of you say yes? How many of you are neutral? So many of you are neutral. So, yes, if you are using the C program, if you are already there in the C, if you are, it is there in this program file, the script, research institute, you will find out of it for. So there you have to go there and select so where we have kept this auto date for and auto we have kept in the desktop folder. So we'll browse it. We'll keep the location, desktop again Autodoc, then auto. What we have to select? Auto grid or Autodoc code? Yes, participants. Yes, we are going to run the auto grid, so we have to select auto grade 4.exe. Open. Okay, so now the parameter file name. So it is also there in the desktop. Okay, so when you click it will give you the information that grid log file will be prepared with the same name in the same location. Just launch it. It's launched. So click on launch. So when you click launch, so what will happen? The window that is running. So we'll wait for some time until the grid so log file is being generated. So it's gone. It means your grid parameter file, grid log file is created. So what you will do, you will go to the desktop and you will check whether it is being successfully completed or not. If it is not successfully completed, then again you have to run the grid. So we are going to check it. Okay, we'll try. We'll try to finish this within 5 minutes. Okay, we won't have much time because it is the lunch time, and if we skip lunch, then it will be difficult to us. So what you have to do, you just click on it, open with Notepad. Okay, here. Sir, please forward. So when you scroll down, you will see all the calculations which are made by Autobot. At the bottom, you will see the composition is there, our rate and successfully given. Now, quickly we will run Autodoc 4. So again, moving to the window, same thing we will do. We will prepare the grid parameter file and we will give the input to Autodoc.exe and it will generate the grid log file and then we will see the effects for the results. So we'll go to docking, and then we will go to the macromolecule and select frigid file name. Our file was stored in the desktop, so we will go to the desktop or Autodoc, and we'll select our 5IVR. Okay, so here is it, and then we will go for docking the ligand, and we will choose the ligand. Our ligand is our info file, and we will select. Okay, it will give you the Autodoc ligand for 4 ligand parameters. You accept these parameters. Then you go to the again docking and the search parameters and genetic algorithm. Then you accept default parameters. Then you go to docking parameters. Then you accept the default parameters. Accept. Then again you go to docking. You go to the other options and add Autodoc parameters. You select accept. Okay, then you go to the docking and you select output and you select the Lemachian genetic algorithm. Just again, we have to save the protein. We have to save our parameter file in the Autodoc. So to go to the desktop, Autodoc, and the docking parameter file, save, and this is the previous file. We are going to replace it. Okay, so then again we have to go and check it whether it is being prepared or not. We will remove the previous log file. Okay. So now we are going to run the Autodoc. So go to the run Autodoc, and then what you have to do, you have to again choose the program path name. And in the program path name, what you have to do, you have to go to the desktop, you have to open Autodoc, and you have to select the Autodoc4.exe. Okay, so this is our program path name, and you have to select the parameter file. Where is our parameter file? Again, our parameter file is there in the desktop. So go to the desktop. You open your parameter file, and automatically it will give you the document file information where it is going to be stored. You just click on launch. We'll go to the analyze function first. So it is written. So it will take more time than the time when you can validate parameter file to be created. It will log file to be. So we'll wait for some time. Meanwhile, if you have any query, you can ask me. Yes, I have two queries. The first query is if about like in molecule quality and it is having a multiple tolerances like more than 16 tolerances, and how you can perform the number. See, if a ligand have more than 60, if we talk about more than 60,000, so we can't consider them as a small molecule. They are proteins. Okay, they are proteins. So you have to go to the other plant and protein talking or a peptide protein talking. So that is a different things. Okay, we are here working on small molecule which have at least or 30 torsions. Spoken words. We don't put it we go to a class three and class three plus the residuals which are involved in the active site. So how we can select those residues? Yes, that I can show you in this workshop. Like we will not hinder the Autodoc because if we do something else, how the processing speed will be reduced and we will not get the results on time. So here you can see there is a plus symbol in the front of our line here. Is it visible? Good. Sir. File ID here. File ID here. So I asked Dr. Robert to test the computer because it is running the Autodoc. So it's sorry for it. So you see the plus symbol in the front of file ID here. When you click on plus symbol, you can see that this amino acid will be visible. Okay. So either you go to the PDBG or you go to the server, you write on the file ID here, it will give you the information about the pockets. You use that information, you select the protein, you select the amino acid from the list. When you select it, what will happen and which format you want to select. Okay, if you are able to select it in the line and stick model, so it will not be here. So what you have to do, you have to change the appearance of that amino acid. So you can change to the balancing method, or you can display, or you can change it to the ribbon structure. Okay. When you change the history or what is the history, the particular amendment is clearly visible to you in the screen. So just move to scroll down the grid parameters, dimensions, and location box. You can take a grid parameter box that has the location, and you can. Okay.
My next question. I was drawing some molecules in Camera, and afterwards I made them 3D file into that code. But after the docking, I found that the structural compound was not this model.
Yes, actually, what happened, you have drawn a 2D structure and you have not optimized the structure from the 12th stage to a 3D structure. Okay,
I have optimized by using the array guidelines.
yes, see, actually, what happens, when you are using the chemistry, you draw a structure in a chemistry, you optimize the structure, the same structure in a 2D format. Okay, the same structure in a 2D format should be optimized first. Okay, then ask the chemistry to convert that particular 2D structure into a 3D structure. So it will convert it into a 2D structure. Now, see the structure in some readable formats or in standard format like mol format or SDF format or it's a or IBM the terms. So it is advisable that you create, you see that not in the chemistry format, you stay in the mol format or an SDF format. Now open the open window GUI and convert the SDF format or mol format into a chemistry format. Similarly, there are a number of possibilities are there. Some optical recognition systems are there, which are based in the smile format, and including the smile format, and in the
Next question. After the talking, I have found that there are two structures that historically, the first point that the structure being broken.
okay.
So in that is what
See, in that case, the problem will be your ligand. The ligand structure is distorted.
Yeah.
Okay. That is the problem. I'm getting that you are using the Pyrex version. You are using about right. See, there are two cases. If a person is using a Pyrex and it is and he or she is trying to open the results in the orthodox format, the main problem is the generation of the ligand. If you have not properly prepared the ligand, it will not give you the proper result. Okay, your ligand will be distorted. You can see the interaction with the other amendments. Your time and the will be very, very less. So these are some problems that you have to consider. Second thing, you will get the results. If you get the results, so only molecular docking is not the way that you should go for the study. You have to prove that whether the ligand, if it is attached to the protein, can withstand the body condition. And if you do this exercise in the computer, it is known as the molecular dynamics simulation. So the molecular dynamics will give you the idea that in what body conditions, and you will have the water, you will have the salts, and you will run it. And when you run it, if the ligand is moved from its active side, it gives you the indication that your document is not proper and your ligand is not attached to your.
In the presentation, what we have shown, that was all the things, but how we can make the complex protein and ligand for
Yes.
for the
Yes, I will give you the answer. But our talking for over now, and the people are very good. So first, we can entertain your question. Let us first complete our session. So here you can see that the video which is showing that your document is ready disappeared. It means that your document is completed. Okay, here our log file is being created, so I will rename it. Dot D
or G. Okay, no problem. I will open this. So I will ask my colleague to please scroll down. So this is our document log file, and if we scroll down, you will find out whether our document has been run successfully or not. So here there is an indication that it is successfully run and it has taken our 110 seconds and the real time is 8 minutes 21 seconds. So we have consumed 8 minutes and one of this token. Now I will open our document to find out the interaction of the options. Yes. So we'll go to the analyze, we'll go to the docking, we'll open it. We'll go to the folder where our folder is there, desktop, Autodoc DLG file. We'll open it. It will give you the information that if you want to give any name, there is no issues with the name. So what you will do, you will click on it. Okay. Then it will give you the information that the 10 doc configuration was formed. You click OK. You can go for analyze. You choose this macromolecule. You choose 5-IDR protein. And you select the macromolecule, you can go to the analyze, and you can go to docking, and you can click on show interactions. So when you click on show interaction, the display will be changed and it will be converted into a light stage. So here you can see there are certain display options are there, whether you want to keep the close contacts or not. Okay, so I am zooming out. So this is some amino acids are visible on it. So what we will do, we'll just go to the analyze, we'll go to the confirmation, we'll ask it to play rank by energy. So when you click on it, it will give you the informations. So here are the information. Here is the and symbol. And symbol. When you click on the and symbol, it will give you the set play option display. You click on show information. You click on build hydrogen bond. So here you can get the information. So I think you people have given the long information build parameter file, right? There is some information that you have given wrong. So here you can see we have certain displays. So what we can do, we can go for analyze, we can go for confirmation, we can go for play run energy. So when you play run energy, you will get the protein, the which are interacted in the read. So again, we will do what we will do. We will go to the analyze and we will go to the clustering and we will ask it to show. When you go to the clustering, you will get the information. So there is certain problem with the grid parameter. I am thinking that there is certain problem with the problem. So when you see that there is a confirmation, the confirmation has been reduced. It is reduced in between 5 to 5.5. But the confirmation, you can see the confirmation and the so it is also an acceptable. Okay, but what we have to do, we have to do on the amino acid, whether it is bind to the same amino acid of AG and BG or not. Okay, so this is all about the order of workshop, and I thank you very much for your patience. And if you have any queries, you can ask me. Online participants, it's very difficult to answer your queries because we are running out of time. And I will entertain a few more questions from the audience who are sitting in the audience. Yes, anyone? Yes. Okay, comments. Okay, sir, I will give you the answer. Let them finish. Both the participants who are coming within the
Sorry for the interruption. I know that the workshop was going great, and I know that this has shared or bestowed wide knowledge in the handling and operating and dealing with the recent free softwares from online. And we have made a small video, descriptive video by our honorable coordinators, Dr. Rahul Kumar Maurya and Dr. Yashir. And we are going to share the same with all of you, those who have been attending us online and offline in the respective WhatsApp group. So you can also download the video, detailed video. And if you still get stuck somewhere, so you can make a call to our honorable coordinators, and they will be definitely available every time to help you with your queries and doubts. So due to constraint of time, I'm sorry enough to declare this session an end, because now we shall move for the lunch. So the delegates are requested to move to the corridor to have their lunch and be seated in the auditorium back sharp and by 1:45 a.m. because we are going to start with our first technical session 1. And the speaker will be Dr. Arsa from ANU. So I just request everyone to have your lunch and be back by 2:45 p.m. sir.
Those who want to leave for a lunch, so please they can, and those who have any queries, I can call the queries. Please, there's girl have some queries. Sir, I'm getting that.
Hello.
I definitely need this.
Because of time constraint, the ATM is.
Just go over question. Yes.
Well, I have used one vegan for my dogging study, and to validate that dogging, I have used decoy also. But while dogging, in literature, I have found that those decoy they generate 50 decoys, and their binding energy binding affinity is less than the ligand molecule. But when I was performing using my ligand, I didn't find so. So I have not included that in my study. But what is the solution, sir?
The solution is you have to read off your okay. The first thing what you have to do with the problem is with the ligand because the people are retrieving the ligand from a different databases. Okay, so first thing what you do, you just download the ligand in a good format which can be easily converted and it should be in the 3D format. Okay, keep in mind that if you are working in a computer and you are not with the simulation, it is very necessary that you should go for the 3D stuff. First, and second thing, if you want to keep your molecular document, as you say, that you have to validate. So validation, for validation, I prefer that for validation, you go for molecular dynamics simulation. Okay, so for molecular dynamics, you need the GLOMAX, which is a freeware. Okay, GLOMAX is free, but number of commands is required to run it. Okay, first thing. Either you use paid software like Studio, or you can go for a testament that is there in the studio. So the testament, I think it is very user friendly, and you put your complex, you input your complex investment or a complex, it will give you the right information. Because in molecular talking, the ligand should be interact with the same amino acid during complete dynamic simulation time. Okay, if you have chosen 100 nanosecond dynamics, so that ligand should, if it is like here, now you can see it is binding to A and B chain of isolation 50. Isolation 50. Okay, so during the complete 100 nanoseconds, nowadays the people are required 100 nanoseconds, 100 nanoseconds dynamics. So after running the molecular dynamics, your protein and your ligand should be the ligand should be attached to the same amino acid, that is isolation 50 for greater than 95% of time. And for my case, more than that, it should be equivalent to 100%. If it is reduced, means it is somewhat leaving the system. And if it leaves the amino acid number of times, then it is not acceptable. Okay.
How can we learn this molecular dynamics and the simulation, sir?
Sir, See, we are expert in molecular dynamics, but it is not a not not a you can't learn it in 1 or 2 hours. You have to give time for it. Number of things are there that know about molecular dynamics. First thing you should know about the protein. Right? It's not only that you know about a protein there, first structure of that, primary structure of that, secondary structure of that, tertiary of that, ordinary. It is not like that. Okay, there are certain geometries between the mind that learning about. Okay, you should have each and everything in mind before learning the molecular dynamics. Okay, so we can help you out, but right now, immediately, our engagements at much that we can only collaborate with your guide so that we can do that particular portion on your behalf, and we can even teach you, but
So do I need to come, or do I need to, consult with my guide and collaborate with,
yes, yes, you can ask the guide.
thank you so much.
I think thank you very much.
नहीं बंद कर दीजिए मेन स्क्रीन चलने दीजिए आ रहे हैं सब लोग खा खा के राइट नहीं funny, ये दो मिनट स्टॉप शेयरिंग